Hello, this is a workshop on multiplex immunofluorescence image analysis with QuPath. My name is Sebastian Gilbert, and I'm an image and spatial omics computational scientist at the Molecular and Advanced Pathology Core Facility. This is a core facility at the University of British Columbia. This workshop was first run on the 30th of June 2026, and this recording is intended for those who are unable to attend, and for a wider audience. The objectives of this workshop are as follows: to set up and navigate QuPath, and to understand the ecosystem that is developed around this free and open source software for digital image analysis. To understand some concepts and key terms from digital image analysis, such as channels, colors, dimensions, and how they relate to a standard image analysis workflow. Then we'll view and interact with digital images and conclude that with quantitatively inspecting images for their quality. This then all relates to how we can then analyze multiplex immunofluorescence images, performing tasks such as tissue and cell segmentation, or cell classification and phenotyping, and then how we can display those results at the end. To achieve these aims, we've broken this workshop into 16 modules. The first part of this workshop will focus on understanding digital images and the concepts that relate to them, all using QuPath. Then in part 2, we'll focus on how these concepts relate to multiplex immunofluorescence images and performing the tasks necessary to analyze them. There is some content that is not included in this workshop, and these will hopefully be addressed in advanced modules. These are the ones that are in the works, and the intention with what we've currently put together is that you'll be able to perform all of the tasks up to the foundational series of data that can then enable you to go further. The modules are named after concepts from 3 main resources. The first from the network of European bioimage analysts, who have a great website going through each terms of from these modules. There is the book developed by Pete Bankhead, the developer of QuPath, talking about analyzing fluorescence microscopy images with ImageJ. And then there is the carpentry lesson called Image Processing with Python, which some of these concepts have come from. Now be careful when reading around the subject that when these terms are used, we'll be using the definitions described by these resources. And so those that from other resources might have slightly different properties associated with them. So what is image analysis and image analysis relating to multiplex immunofluorescence images? Well, multiplex immunofluorescence images, or MIF images, involves analyzing multiple fluorescent signals within a digital image and extract meaningful data. Some key aspects that we might want to consider are spectral unmixing. This is when a multiplex immunofluorescence image is acquired that we want to distinguish the different wavelengths of fluorescent signals to accurately quantify the markers that we're interested in, measuring the biology we're interested in. We might want to extract the intensity of those measurements, evaluating the fluorescence, which then indicates something relating to the underlying biology. Image analysis also considers automation and how we can use software and computers to be able to automatically visualize, identify, quantify, and measure our images. And then how can we understand the spatial relationship of these different fluorescent signals in the biology we are interested in. So the standard image analysis workflow often looks something like this. we have 4 different steps, where we have the first is image pre-processing, where we're trying to increase the signal from our fluorescence markers and trying to remove any background or noise from our images. You'll often hear the term signal to noise ratio, referring to the quality of that image. Then in segmentation, we're trying to identify the different features of our image and the features that relate to the biology of interest. After which we then can merge the features into objects which we can then manipulate, measure, and maybe perform some quality control through post-processing. And then finally, measuring the final objects of interest and performing some kind of quantification and comparison between the different types of tissue or different conditions that our experiments have been put through. So how does this relate to multiplex immunofluorescence images? Well, in this workshop, we'll be focusing on this workflow, which is distinguished as these 6 steps. For example, in this image of a tissue microarray, we have a single core. We first want to perform quality checks to make sure that the image is of a good enough quality to pursue our analysis, but also check if all of our markers have been successful. And this then refines our image to the channels that we are able to pursue with. Then, performing tissue segmentation, we identify the areas that we would like to analyze and maybe identify other compartments of the tissue. While that step can be automated, following that we can perform artifact removal, usually a manual process with maybe some help with automation. From there, we can refine our tissue area and then perform cell segmentation, where individual cells are identified within those tissues or tissue compartments. Those cells are then classified or phenotyped, and then this is what we can measure as our cell composition analysis and compare between the different tissue compartments or tissue areas. Now, following that, this provides the foundations and objects to perform different spatial analyses. For example, we can perform spatial statistics. We can identify our different cell as part of a network. Introduce concepts such as niches. And maybe perform and analyze our images using tools available in the literature, such as cellular neighborhoods. Now, these are dependent on the specific questions and hypotheses being asked about the images, whereas this workshop will get us to cell composition, the objects which can then go further depending on those questions. A little about me. So I started my PhD back in 2020 and was looking into complex spatial structures in biomedical images. This was at the University of Birmingham, and I performed this work co-funded by AstraZeneca and worked with a series of academic collaborators. Following that, during a postdoc, I was looking at developing those methods into commercial tools, before then becoming an image analyst at a core facility and have been since. This has meant that I've worked in multiplex immunofluorescence images with a number of academic and industrial partners. And as a result, have published a series of publications on various digital image analysis tools. I'd like to acknowledge that this workshop is funded and with the help of the Molecular and Advanced Pathology Core Facility, and was initiated at the Birmingham Tissue Analytics Core Facility, where I was able to practice this workshop for the first time. I would also like to acknowledge that the University of British Columbia Vancouver Point Grey campus is located on the ancestral and unceded territory of the Musqueam and Squamish and Tsleil-Waututh nations, and that you can find the materials for this workshop in the links that are in the video description. So that has concluded the overview of the workshop and what will come, and our next module will be installing and navigating QuPath. I hope you can follow along for more.